So thank you very much. Good morning, good afternoon, good evening. I welcome you to this Innovaderm sponsored webinar. We have attendees from everywhere across the world. It's very exciting. I'm Anne-Marie Gaulin. I'm the Chief Business Officer at Innovaderm, and I will be your moderator for the next hour. So today we will present how to handle missing data when conducting clinical trials in dermatology. The presenter from Innovaderm will be Dr. Bissonnette and Ms. Pina D'Angelo. So Dr. Bissonnette is board certified in dermatology, is the president and founder of Innovaderm Research. He has published close to 200 articles and book chapters and give lectures on various aspects of dermatology at various national and international meetings. Dr. Bissonnette earned his medical degree and completed his residency training in dermatology at the University of Montreal, then completed his basic and clinical research training at McGill University and at the University of British Columbia. He was an associate professor of dermatology at the University of Montreal until 2004. Ms. Pina D'Angelo holds a bachelor's degree and a master's degree in mathematics and statistics from McGill University in Canada. She has worked in the pharmaceutical industry for more than 25 years. In her most recent role, she acts as a vice president of biometrics at Innovaderm Research. Pina has extensive experience in many therapeutic areas in clinical trials, ranging from phase 1 to phase 4 studies, as well as PK and PD and bioequivalence studies. The main topic of her recent research include handling of missing data, outlier consideration in vitro permeability testing, and non-parametric approaches to testing patch adhesion, irritation in dermatology studies. So before starting the presentation today, please allow me to give you a quick introduction on Innovaderm Research. Innovaderm is a full CRO specialized in dermatology, supporting sponsors in their clinical development from phase 1 to phase 3. We currently have operations in North America, which include US and Canada, as well as in Europe. For trials requiring a global reach outside of these two regions, we work with well-established partners. Our key strengths include the medical, scientific, clinical expertise we bring to study design and protocols, the relationship we've built over the years with our dermatology sites, and operational execution that allow us to provide quality data on time. In 2021, we are proud to have won the CRO Leadership Award. Elected by biotech and pharma industry, Innovaderm earned a top spot in the capabilities category and was a top runner-up in the expertise, quality, compatibility, and reliability categories. In the past 8 years, we have supported our client with more than 100 trials with systemic and topical compounds, ranging from phase 1 to phase 4. Our experience varies from most research indications, such as atopic dermatitis, psoriasis, alopecia, and acne, to more challenging indications such as palmoplantar psoriasis, hidradenitis suppurativa, notalgia paresthetica, prurigo nodularis, and so on. I have a question for you. You can go to the next slide. So we will have a different section during the presentation. We'll have poll questions that will pop up to you on your screen. So please answer these questions. They're anonymous. So the first one is, how important are biostatistical capabilities when selecting your CRO? So that will just give us some marketing indication. It's always interesting. And Pina will have some in her section as well. So without any further delay, let's get going with the topic of our webinar, how to handle missing data when conducting clinical trials in dermatology. Let's welcome Dr. Robert Bissonnette and Pina D'Angelo. Thank you very much.
Good. So thank you very much for this introduction. My name is Robert Bissonnette. I'm the president and founder of Innovaderm Research. I'm a dermatologist. I'm not a biostatistician, so I cannot give you a talk on how to handle missing data in clinical trials. However, I can give you a clinician's perspective on this interesting and important topic. So one key question is this important? How important is handling missing data in clinical trials. It's important for biostatisticians, obviously, it's important for FDA, but is it really important from a clinical perspective? Next slide. So in the next 5 to 10 minutes, I will give you my perspective on the importance of missing data in clinical research. I will share my experience in terms of why are data usually missing in trials, and I will also provide some suggestions in terms of avoiding missing data. Today, the focus will be on how to handle missing data, but obviously the most important point, in my opinion, is try to avoid missing data. What can we do to try to avoid missing data in order to have to use those methods, but to be in a situation where the method may not be that important in terms of results. Next slide. So this is an example of a very recent publication on Bimekizumab, which is a biologic for psoriasis. One of the most efficacious, if not the most efficacious systemic drug for psoriasis. In these phase 3 trials, 435 patients were randomized to receive either placebo or Bimekizumab. The non-completer rate was extremely low. So at week 16, only 3% of patients did not complete the study. So psoriasis, as most of you know, is a fairly stable disease as opposed to other diseases in dermatology, which are much more fluctuant. It is a symptomatic disease, but it's not as symptomatic as others. And in this trial, the completion rate was very, very high. Next slide. Now, these are the results of the primary endpoint of the study, IGA 01. Only 1.2% of patients randomized to placebo achieved an IGA 01 as compared to 92.6% of patients randomized to active treatment. So from a clinician's perspective, when I look at this, regardless on how the missing data is handled, the results first will be highly statistically significant. And to me as a clinician, knowing that 3% of patients did not complete the study and there's missing data for other reasons as well, if in the end, the IGA is 95 or 96 or 88 or 89 or 93, it doesn't really make a difference. I won't prescribe the drug more or less. To me, this is super efficacious. So I would say that in this situation, the way missing data is handled, to me as a clinician, is not extremely important. However, next slide. We shift gears and we're looking at a completely different indication, atopic dermatitis. This is the phase 2 study conducted with Abrocitinib and atopic dermatitis. Moderate to severe patients, this is a very symptomatic disease. Patients have intense itch, they don't sleep. In this study, patients were randomized to 4 doses of Abrocitinib or placebo. The non-completer rate was very high, 41%. Now, 41% is more than 10 times higher than 3%. So handling of missing data in this trial with about 50 patients per group becomes important. Next slide. Luckily for Pfizer, the drug was extremely efficacious. So the 100 milligram and the 200 milligram groups were statistically significant from placebo. The difference was very, very high. And in the end, the way this is handled from a statistical perspective would probably not have changed the primary endpoint results very much because of the high efficacy of the drug. A third and last example. So recent data published on a completely different drug, still an atopic dermatitis. So remember the fluctuant, very symptomatic disease where patients were randomized to either Etrasimod or placebo. Primary endpoint was at week 12. Next slide. And now you see that at the primary endpoint, there is a numerical difference between the active groups and the placebo, but the difference is not very big. And looking at the error bars, you can see that it was not statistically significant for that primary endpoint. However, when you look at the change from baseline, in EASY, one thing you see that is very different from the previous trial is that the efficacy does not plateau around week 4 or 6, like with the JAKs. It was still going down even at week 12, probably because of the mechanism of action of the drug. Next slide. In addition, there were issues during that trial where patients, where a drug was interrupted in patients where it should not have been interrupted. And this was close to 20% of patients in the highest group. Slide. So when the company did a post-hoc analysis, excluding those patients, then the curves separate very well and it becomes statistically significant. So in this example of a phase 2 study, smaller sample size, efficacy not as high as what I showed you before, maximum efficacy probably not reached by week 12, then handling of missing data can be very important. Now, why are data missing in clinical trials? Next slide. In my experience, the number one reason why data is missing is early term or loss to follow-up. So patients will decide not to complete the study. Missed visits. So these are patients who would miss a visit. It could be an interim visit. It could be the primary endpoint visit. Obviously, this is very sad. And this could be for various reasons. It could be that patients are not available. Could be that the site is not available. We had a few of those during the COVID-19 pandemic. Sites closed, for example. Use of rescue prohibited medication. So this could either be a case where we need to decide what to do with data that is present, or it could be a case of data that is not collected because from a protocol perspective, after rescue medication is started, efficacy evaluations sometimes are not collected. Problems with e-tools. So nowadays, most of our trials use e-tools, and it's not rare that we have issues with e-tools. Either pads or tablets we give to patients. It could be e-tools that we're using on site when the patient comes in. If the device doesn't work, if we cannot have access to an option, or if we cannot solve the problem fast enough, then we end up with missing assessments. And sometimes sites just forget to do assessments, and they're missing because the patient left without having all the assessments completed. What can we do? What can the industry do? What can CROs do to try to avoid missing data? A few ideas. First, open label extension is very important. So in the first trial I presented, the trial with Bimekizumab and psoriasis, there was an open label extension. Patients knew that if they were able to complete the trial, then they would have access to drug afterwards. In the phase 2 Abrocitinib data, it was not the case. And this is often the case in phase 2. Unfortunately, often open label extension is not offered. So if a patient after a few weeks on treatment doesn't see improvement, they might say, well, on a placebo, the drug doesn't work. So I'll quit the study because if I'm on placebo, I won't have access to the medication. This is more important if the disease is symptomatic. So for example, alopecia areata is an indication where we've been active recently. This is a disease that has a tremendous impact on quality of life. However, the skin lesions don't burn, they don't tend to itch. So from a symptoms point of view, patients don't have significant symptoms. So dropout rate in alopecia areata study are significantly lower than in atopic dermatitis, where patients have intense pruritus, they cannot sleep, and they tend to get discouraged a little faster. Even a short-term open-label treatment makes a difference. So if patients after the placebo control period can have access to drug for a few months, that really helps and makes a difference in terms of minimizing the non-completers. Be realistic in terms of data collection. More and more nowadays, we see studies where what we ask patients is really a lot. They have to fill 5, 6, 7 quality, 10 quality of life questionnaires. We're asking for biopsies and transepidermal water loss and tape stripping and PK, and they need to stay on site for hours after drug is administered, either to do PK, to do safety evaluations, post-topical applications, evaluation of efficacy, even when the patient has been on drug for a few months. So all this taken together, what happens sometimes is patients just get discouraged because the visit will take half a day or even more at the clinical site. Next slide. Patient e-data collection, as I said before, this could be an issue, could even be a major issue. We see more and more studies where this is extremely important. It's either a key secondary endpoint, like pruritus and atopic dermatitis, and sometimes it's even the primary endpoint, where we're looking at a drug, for example, that targets pruritus. So key things are to use vendors that are experienced in the indication and assessments. If they program something for the first time, chances of having an issue is higher. Use vendors that are known to the CRO. So a bad combination is a new vendor unknown to the CRO who is working in the indication for the first time and programming in the first time. And be realistic in terms of what is required from patients. So pruritus NRS on a daily basis for a few weeks, it's fine. For 3 months, it's long. For 6 months or a year, it's not really realistic, in my opinion. Obviously, part of the study, according to expected missing data, as I showed you, is very different from one indication to another. Rescue therapy is also something that is key, specifically in indications like atopic dermatitis. Many protocols tend to offer rescue therapy. Some protocols will offer rescue therapy as early as day 1, so immediately after randomization. Other protocols completely prohibit rescue therapy. This was the case, for example, of the phase 2 Abrocitinib study. So patients were not allowed to use rescue therapy. From a data perspective, it's good because the data is not mixed up with too many patients using rescue therapy. However, the dropout rate is higher. So I usually suggest to establish a time where rescue therapy is not allowed, because if you allow it as early as day 1, what you sometimes see are patients coming in, patients read the consent form, they know they can have access to rescue therapy, they randomize on Monday, they come on Tuesday and they say, well, I want rescue therapy. It's in the consent form, doctor, I want it now. And then you lose a patient for your primary endpoint probably, or be considered as a non-responder. It's also very important to make sure that you define the population on which the efficacy will be estimated, and you specifically mention in a protocol, or at least in the SAP, how these will be handled in terms of statistical approach for patients who use rescue medication. And I think this was my last slide. If you have questions, we'll take it at the end.
Yes, so just before Pina starts her section, you have a button on your screen or on the top or at the bottom that's called Q&A. So please feel free to send in some questions, and towards the end, I will be able to run them by our speakers. Thank you. Pina, your turn.
Very good. Hi, everyone. Thank you for joining. It's always very exciting to see people interested in biostatistics, even though I'm sure Dr. Bissonnette's presence helped. Next slide, please. Just as a quick outline, we're going to look at the different types of missing data. We're going to just take an example of a case study. We're going to talk about single and multiple imputation sensitivity analysis, and then wrap up with a discussion. There's 2 things that I think I'd like everybody to retain today, because I appreciate not everybody that is with us today are statisticians. I think I'd like everyone to try to understand the different types of missing data. And the second thing would be just to learn the basics of the statistical methodology that is used for imputing missing data and conducting sensitivity analyses. When it actually comes to doing those imputations or conducting, those would be the statistician amongst us that would do it. But just to get a basic understanding, that would be great. And if you have any questions, you can always let me know. Okay, so different types of missing data. I hear a lot of times as a statistician, people will say, what's the best way to impute my data? How was the best way to impute mine? And we have a lot of focus on the method of imputation. What I recommend is you have to understand how and why your data is missing to begin with. And then you can choose the methodology that is best going to suit how to take care of that. So we generally have 3 categories in statistics: missing completely at random, missing at random, and missing not at random. So many times you'll see MCAR, MAR, or NNAR. So we'll start with the first one, which is missing completely at random. So this one is pretty straightforward. It pretty much means what it says, which is there's absolutely no relationship between whether a data point is missing and the value of what that would have been. So they're completely random. So example of this would be a questionnaire might get lost in the post, or a blood sample might be damaged in the lab. So this is completely random. It has nothing to do with the treatment that was administered, and it has nothing to do with what the outcome would have been. In this category, so if data is missing completely at random, what we call throwing out cases, which is, some of you might have heard of it as a observed case analysis or a completed case analysis. This is generally an acceptable method that does not bias your statistical results generally. I will say though that it is very rare in clinical trials that you can say that all of your data is missing completely at random. So this is not a very strong assumption most of the time. The one that I think most of us tend to default to is this one, which is missing at random. So it means that the propensity for a data point to be missing, so the probability of you having missing data, is not related to the missing data itself, in other words, the value you would have measured, but it has to do with something else that you can observe in the data. So an example of that is men are more likely to tell you their age or weight than women. So if you are asking a questionnaire and you have missing data for men or women and it pertains to their age or weight, chances are it's not because of the actual value that you're trying to assess. It has to do with the gender. Men are also more likely to drop out in clinical trials than women. Men are less likely to fill out a depression survey. So again, this has nothing to do with their level of depression after accounting for the fact that they are male. So the key here is after accounting for whatever variable you think caused that missingness. Another example is if a child doesn't attend an assessment at the clinic because they are ill, and I did put in bracket genuinely ill, this might be predictable from other data that you have from the child's health, but it would not be related to what we would have measured had the child not been ill. So some say that deletion is fine, but only if you are able to control when you're conducting your statistical analysis, if you're able to control for these variables. The more common approach to use would be a multiple imputation, because the deletion does introduce bias. So if you are going to use an observed case when you know that you have missing data that is missing at random, it may not be the most robust solution. Next slide. The last one is my little bandit here, which is the little devil, missing not at random. So this means that the data that is missing specifically is related to what you were trying to measure. So an example of this is a person doesn't attend a drug test because they took drugs the night before. So the outcome that you're trying to measure, which is, does the person have drugs in their body, yes or no, is directly related to the fact that they did take drugs the night before. So if you were to just exclude that data, you can see how quickly you would bias your results. Another example would be that patients with worse health were more likely to have missing questionnaires. So if you completely disregard people that have missing data when you know that they might be the iller patients, then this again could lead to a lot of bias. The reason why we call this one the bandit or the most problematic case is that we don't have that many methods of imputation that say, okay, in a case of data not missing at random, I can do this. We don't have that many. There are very few, if none actually, methodologies that will really account for this. What we try to do is we try to include as many predictors as possible so gender or other things that might tell us why that data is missing, in order to get closer to a missing at random model as opposed to a missing not at random model. I'll talk a little bit more about this when we get to the sensitivity analysis. Next slide, please. So let's just look at a case study. We'll look at a hypothetical atopic dermatitis study with N equals 92 patients. We're looking at a parallel design, one-to-one randomization, active versus placebo, so very straightforward. We have assessments that are performed at baseline, week 1, 2, and 4. So we have a repeated measures design across time. And our primary endpoint is the proportion of subjects with an IgE score of clear 0 or 1. So at week 4. So we're looking for 0 or 1 at week 4. And the statistical test that we will use is a CMH test, so a Cochrane Mental Hensel test, stratified with baseline IgA as a response, as a stratification variable. Next slide, please. So let's say this is what our results look like. So we have at week 4, we have the percentage of patients that scored yes and no for scores of almost of clear or almost clear. And we see that our p-value is 0.038. So this is a phase 2 study. We look at this and we say, oh, rather encouraging. And now, of course, for those of us in the phase 2 versus phase 3 world, we start to think about, should I bring my drug into a phase 3 study? So normally, next slide, please. When we see these results in general in phase 2, what I always suggest to everyone is, understand how would missing data impact your study, for example. In other words, how robust are your analyses or results from the phase 2 study? So I'm just going to take a random example just to prove a point of how quickly you can get missing data to be important in your study. So let's just randomly select and remove 2 patients or 4 patients or subjects. So that represents about 4% or 9% of the data of your study, which is not atypical for clinical trial studies. Next slide, please. So here what we did is we took our original results that had that nice p-value of 0.038, and we essentially randomly set 2 subjects per treatment that didn't reach week 4. So 2 subjects per treatment that didn't have an IgA score response variable at week 4. So you see how quickly you have p-values that are now not so interesting. And I say not so interesting, so you're at, a little bit over 0.05 in the first case and, probably double that in the second case. The reason I say not so interesting is not from a statistical perspective. Statisticians will look at this and take these things with a grain of salt because you still are under your 10% and close to 5% significance alpha nominal level. But I think many of us from a client perspective and from a brand perspective would have to explain this, for example, to board of directors that are looking to invest more money in a product and decide whether to bring it into a phase 3 study or not. So here's when it becomes a little bit trickier to try to understand how missing data impacts the true result of your study. Next slide, please. So I'm sure a lot of us have heard of last observation carried forward. So, we're going to look at that and say, well, what would happen if I took those missing data and I did an LOCF? What would the results look like? And I just want to go back to the first case that if I would have examined my data and the missing data after consulting with clinicians would be that, oh yes, this data here was missing completely at random, then you would be able to justify the observed case analysis, which is I just used data that I have. But again, it is really very rare that it is certain that you have data that is missing at random, completely at random. Especially in a case like the one that I just described, you really would have no way of knowing, short of every single person that, people that your patients that are missing said, oh, it's because, I just decided to go vacation in Florida, for example. Then maybe you can make a case. But otherwise it would be really challenging. So let's take a look and see what the results using an LOCF method would be like. So here we see that, okay, we're still not quite at the 5%, but it is, this is using the full analysis. So we have carried forward the observation that was last available. The point here again is the LOCF, which we all, well, I shouldn't say we all know, but I will state that the guidance that we have from the regulatory bodies like Health Canada or like the FDA don't really favor an LOCF method very blindly, generally speaking. Certainly not as a primary analysis, and we'll get into that in a little bit with respect to sensitivity analyses. Next slide, please. So let's look at just different types of imputation. So a single imputation, this is kind of the easiest case scenario. So many of us, we've looked at last observation carried forward. So that is one method of single imputation. In other words, you're just imputing one value for the data set that's missing. Another one is a worst observation carried forward. So you would take the worst observation that you see across baseline week 1 and 2 and carry that forward. Normally, when we talk about a worst observed value being brought forward, that is probably the baseline value unless a patient got worse. So what you're automatically doing is imputing a non-responder most of the time in this type of an imputation. You'll have other imputations that look at a simple mean imputation. So what you essentially do is, let's say you need to impute a visit 4, then you will take the mean of all of the data points that you have across the subjects. Maybe you could do it by treatment and impute the mean that you are seeing, and that would be a mean imputation. I will say that this one is also, it has its limitation in terms of what we call a reduction in variability, because you're essentially imputing a mean, so you're artificially decreasing the variability of what could have been the spread of your data. In general, the validity of a single imputation depends on specific assumptions, again, of the missing data. So in certain cases, it's okay to do an LOCF, and in certain cases, it's really not. I don't want to talk too much about estimates here, but I do want to open the door for the statistician amongst us. The FDA guidance on estimates will usually guide us very, very much in how to define our estimate. And in certain cases, in certain of the categories, it is actually okay and recommended to use an LOCF approach. The single imputation, the assumptions are often unrealistic, and therefore they'll potentially bias your results. So they need to be taken with, quite a bit of caution, in my opinion. Next slide, please. So multiple imputation. Multiple imputation actually has been around for a long time. It originated in the early 1970s, and it's gained popularity over the years. So when we talk about an imputation, right, it generally refers to one set of, I'll call it plausible values. In other words, one set of estimated values that you can impute for your data set. So that's the single imputation. Multiple imputation essentially represents multiple sets of values that you can impute, hence the reason multiple imputation. So what is multiple imputation? Because a lot of us have probably heard about multiple imputation. It's not that straightforward, but it is once you understand what it's doing. So there's 3 steps in a multiple imputation. You have your imputation step, you have your analysis that is done on each of the imputed cases, and then you have your pooling, because you want to pool across all of the analyses that you get. So let's start with the imputation step. So missing values are identified and are replaced by a random sample of plausible values. So you'll start off with 1, and you essentially impute values so that you have a completed data set. And you do that, for example, 25 times or 50 times. Normally 25 times, 25 data sets that you would impute are preferable to reduce sampling variability from the imputation process. More is better. We generally see, however, that the data does converge very close to 50 data sets. So what you have are essentially 25 of your original data sets with different values that you imputed for your missing values. Then your second step is you're going to, for each of the 25 data sets that you constructed, you're going to run your primary analysis, be it a CMH test or be it an MMRM or an ANCOVA or an ANOVA. So you're going to have 25 results. That's the second step. And then the third step is what we call the pooling step. This is the one where you combine the results from the 25 analyses, and it gives you your final results for multiple imputation. So of course, I've explained this in very simple terms. There are many items to control when you're doing an imputation on the statistical end, which would involve the monotone patterness of what your missing data looks like. There's multiple things that you obviously need to take into consideration, but the concept is a pretty straightforward one. So a little bit on multiple imputation with respect to dermatology studies. So dermatology studies often have dichotomous primary endpoints and endpoints in general. So for example, IGA success, which is what we just saw. So are you a responder or not? EASI 50, did the patient reach at least a 50% reduction from baseline in their EASI score? Or a PASI for psoriasis studies of PASI 75, did the patient achieve a 75% decrease in terms of their baseline values for PASI? So these are 0 or 1. Many times I would say that where a lot of the literature will talk about multiple imputation with respect to continuous variables. So if you're doing an MMRM, if you're doing an ANOVA or an ANCOVA, then we often, this is how these methods originally were derived. However, when you're talking about dichotomous data where a CMH test is or a logistic regression is used, then it needs a little bit more attention, so to speak. And so there's actually a lot of work that has been done and has been published on this topic. Again, I encourage you to contact me if you do need any help with this. But essentially, the chi-square distribution is highly skewed for smaller degrees of freedom, and thus obtaining the results, the combined results of a CMH test from an MI requires a transformation that normalizes that CMH statistic. So you're doing a normal transformation to the CMH. So for example, the Wilson-Hilferty transformation is well known and is something that we definitely use for multiple imputation for dichotomous variables. Next slide. Okay, so a poll question. Have you ever heard of a tipping point analysis? Okay, I'll answer that in a moment. Audrey, I'm not sure how much time we're supposed to give, but we're ready to go to the next slide whenever you are. Okay, so do I answer the question now or later? Okay, I'll answer the question now. The answer is no. A tipping point analysis, or actually, I'll carry on this way. So a sensitivity analysis, there's a lot of options to choose from. Often it's best to conduct the multiple sensitivity analysis just to see if you have all roads leading to Rome, so to speak. So your results might be different. You might get a p-value that's 0.01 and then 0.02 and then 0.049. But if all the conclusions are that you are statistically significant in all of your sensitivity analyses, this really builds a robust case to the to yourselves and to us, as well as to the regulatory bodies that you're trying to make your case for a drug on. So I'm just going to talk about a couple of sensitivity analyses, and there's a lot of them. Best, worst, worst, best, and tipping point analysis. So the best, worst analysis, this is the one where what you do is the missing data that comes from subjects that are in the vehicle or placebo group, you would impute or you would set them to the best possible response. So you assign them an IGA success. And those that are on the active group, the opposite. So you give them the most undesirable. So you would impute them as a non-responder. So this really gives you what we call the best worst scenario. Then there's a worst best, which is the complete opposite. So in other words, you're going to impute all of the missing data for the vehicle group or for the placebo group to be not a good response, so a failure in the case that I was talking about. And the opposite for people that are on active treatment group, you would impute them with a responder or the most beneficial rate. And then there's the tipping point analysis. The tipping point analysis has really gained a lot of popularity. So for those of us that have heard of it, a tipping point analysis is a method of sensitivity testing under the assumption that your data is not missing at random. Or it's missing not at random. So this is something we actually use if you want to understand if my data is missing not at random, this is the sensitivity analysis that you would look at. So I'm just going to give, again, going back to our hypothetical case, what's nice about the best, worst, and the worst best, and I encourage all of our clients and people to do this in general, it gives you a nice range. It gives you what the best possible scenario of your results is going to be, and it's going to give you the worst one. So in a perfect world, if you have the best worst and the worst best results that are all giving you same conclusions, this is a good thing. If in our case here, so the first one, we looked at, I'm looking here really at the subjects that, removing 4 subjects, so about 9% of missing rate. If you look at the patients that were on placebo, impute them all with a responder and the opposite for active, then you get a p-value of 0.447. So this is probably, telling you the worst case scenario in terms of your drug. And then the best case scenario in terms of your drug having an effect over placebo would be when you do the opposite. So you impute all of the missing data from patients who received active as responders, and then you see that you have a highly significant p-value. It's not uncommon to see results like this, especially for a phase 2 study, because you are spanning the globe, so to speak. So you're going from a very best case scenario of having something pretty significant to a very worst case scenario of having something not so significant. Next slide. So I'm going to talk about tipping point analysis. Hopefully I'm going to wrap this up in about 5 minutes to leave time for some questions. The tipping point analysis is well liked by the regulatory bodies, and I personally really enjoy this method of sensitivity testing. It's a nice way to stress your data. So the tipping point approach, what does it do? It's like a progressive stress testing to assess how severe your departures from missing at random must be in order to overturn the conclusions from the primary analysis. So what does that mean? If you have a p-value that is significant for your treatment, you're going to stress your results and say, okay, how does my active treatment, my group that received the active treatment, how bad do they have to be before my p-value becomes non-significant? So what we do is we use a shift parameter to achieve this. And we keep shifting the parameter until the results tip. So tipping means you go from significant to not significant. So the tipping point approach is not intended for a primary analysis, and it's actually a method of sensitivity testing. Next slide, please. I'm going to give an example here. It's actually a textbook example. So just to illustrate a point. We're going to look at a change from baseline in EASY. So I'm going to look at a continuous endpoint this time. And you have an ANOVA or an ANACOVA model that looks at just treatment, visit, and maybe a baseline EASY score here. And all of the other variables, so we have some missing data at week 4, but we have all of the other variables that are present. So we know the treatments, and we have baseline for all of the other patients that had not missing data. So we'll go ahead and we'll, under the MAR assumption, we will run a multiple imputation, and the pooled results from our multiple imputation gives a p-value of 0.0011. And let's say that this is a phase 3 study. So you're like, oh, wow, this is super great. My product can go on the market now. But these conclusions from a multiple imputation, they are based on the assumption that the data is missing at random. So if it's plausible that for the active treatment group, the distribution of the missing easy responses have a smaller reduction than the corresponding distribution of the observed primary responses, then the conclusion of the MAR assumption should be examined. What that means is if the data that is missing, for whatever reason, looks too good from the example based on the observed data than what it really should have been, then your assumption of MAR is not quite right. So you need to do some sensitivity testing. So next slide, please. We're going to use a shift parameter. So, what exactly is a shift parameter? The imputed change from baseline in EASY for active treatments. So you look at only your active treatment response and you make it worse a little bit at a time by adding a delta. You make it worse and worse and worse and worse. And so the delta is K times the treatment difference observed at each visit. So you're going to keep imputing and imputing and imputing and making your active treatment arm worse and worse and worse until you tip. Now, once you get to that tipping point and you say, okay, here my data will go from a p-value of 0.001 to a p-value of 0.0501. That's when the biostatistician is not going to be enough to be able to make a judgment call. And I would probably call Dr. Bissonnette or other clinicians and ask for their input to say, okay, my p-value becomes not significant at this point in time. How likely is it that my active treatment response is this much? And that's when you make a clinical call on how plausible that is, and that will better defend your original study results. Next slide, please. So typical results from a tipping point analysis would look something like this. So you have your p-value that is on the y-axis, and you have your shift parameter that is on the x-axis. So here, if we're using, again, 0.05 as our alpha, then we see that the shift parameter of about 1.5 or somewhere around there is when we go from conclusions of the study being significant to the study being not significant, right? Next slide, please. So the study conclusion under the MAR is reversed, we call it, to not statistically significant at the 5% alpha level when the shift parameter is, in this case, minus 1.5. Now here's the question. If this shift parameter, the imputed change from baseline in the EASY scores using that shift parameter means that subjects that were given the active treatment, those subjects, their EASY baseline, change from baseline scores need to be half of the effect that you saw in placebo. So let's say I say when my results shifted or tipped, I would need to see the change from baseline in EASY in the active group being half that of placebo. Do you think that that would be a plausible situation? Yes or no? You can also say, I don't know, but please don't say that. I'm just kidding. Okay. So I think, next slide. So if the answer is no, like in other words, it would be crazy to have to go from a change from baseline in terms of active response where my change from baseline is half that of what I'm seeing in placebo, then the conclusion, then you say, no, this is not plausible. And your conclusion that you originally made from that p-value of 0.001 that was under missing at random is likely, and your original results are robust. So this is a good situation. If they are plausible, then it's not so good. And for those who work with me, they know that this is my favorite little baby, courtesy of my colleague, Eric Harding. Next slide, please. Okay, I'm going to really wrap up here. So just to echo what Dr. Bissonnette said, really the best way to handle missing data is to avoid it. So what you want to try to do is, as much as possible, set up your studies, set up your protocol so that you minimize the level of missingness in your data. Because the truth is that missing data will always be a limitation when interpreting clinical trials. It's not possible to rub a crystal ball and know exactly what it would have been. And most of the time it will result in a loss of power under any pattern of missingness. Next slide. Handling of missing data validity is an important yet difficult and complex task. So it's not always easy to try to figure out what the best way to handle things are. What we typically do, however, is we try to circumvent this by using sensitivity analyses and conducting multiple sensitivity analyses that all hopefully give us the same conclusion. So when data is missing not at random, which is our little bandit devil, we don't have any methods really that appropriately handle this. However, like I said, if you do construct your best, worst, and worst best, you'll get an idea of what the range of your uncertainty is. Tipping analysis, very, very strongly recommend that you would conduct such analyses in your studies to see how severe departures from missing at random, how much they need to be in order for your original conclusions to be overturned. And lastly, again, to echo kind of, it's always nice when the statistics and the clinicians kind of marry together, is that in certain studies, it's going to be much more important than in other studies. And I would say that in phase 2 studies, because your sample sizes are not really super well powered, it probably becomes especially important to decide how you handle missing data, compared to, for example, a phase 3 study where your data is properly powered, and then it becomes more a question of sensitivity analysis to support your original conclusion. And I think that is it to me. Yes, I did want to give a special thanks to my colleagues Jean-Pierre and to Gabrielle for helping me to come up with some of the results for this presentation. And I will encourage, we are a growing team of biostatisticians and SAS programmers. So anybody that does want to join our biometrics team, don't hesitate to reach out to me and have a conversation with me.
So thank you very much, Pina. So I just want to let everybody know that a copy of the recording will be placed on our website. And that we will follow up with you, all the participants, we will follow up with you in the next coming days. And if you do want a copy of the presentation, we will be able to send you one upon request. So we have time, I think, for a few questions. I have one here. Do we need a certain number of patients to apply multiple imputation? Pina, this is for you, I believe. You're on mute.
Yeah, I just wanted to unmute myself. That is an excellent question. Normally, multiple imputation, obviously, if you have small data sets, then you don't have a lot to simulate from. So I would say that, as a rough rule, you always want at least normality as close as possible. So let's say you were to use a sample size of 30 per treatment is what I would say that you would be wanting to do. Anything under that, again, this is just my opinion on a rule of thumb. Anything under that would probably give you results that, you don't have a lot of room to simulate from.
Thank you. In the hypothetical case study, when talking about best, worst, and worst best, is there a reason why we randomly selected 4 subjects representing 9% worth of data to draw? Is 9% the norm in such analysis?
Yeah, no, so it was completely hypothetical just to see I was trying to see at which point that data will tip, for example. And so it was completely random. What I would say that it's not, under a 10% dropout is quite normal. In terms of a dropout rate that we would power a study for. So we typically will come up with a sample size calculation and then power for about a 10% dropout rate, anywhere between 5 and 10. So, and it is, typically what we do observe between 5 and 10 in terms of a dropout rate. So that's the only reason I chose those numbers.
Very good. So how much percentage of data missed is acceptable in a clinical trial for a good statistical analysis?
0. No, not 0. So, you know, if you look at literature and if you look at textbook, anything that has 40% or more, anybody will say that you pretty much don't have any case to make. So if you have more than 40% or more of data that is missing, you don't have, you know, I don't think you have a very good case to make trying to impute, you know, almost half of your data. I would say that anything up to about 20%, and even 20 is a lot. So I would say 10%. 10%, you've got good, robust methods, and you have good methods that you can use simulations on. Again, these are just rules of thumb because again, if all of my 40% of missing data comes from data that is missing completely at random, then the rest of the data that I have that is representative of what my overall study results would have been, I can go ahead and if that's, you know, if that p-value is significant, you can make an argument for, well, 40% of my patients decided to go to Florida, then, you know, you can make that argument. It really depends on how the data is missing. But assuming that your data is missing at random, so it's not missing completely at random, I would say up to about, you know, 10% is a good number. When you start getting 15 and 20, it's a fair bit of imputation. So in my experience, that's when the agencies will start to stress test your data a little bit more appropriately. So,
very good. So we have 3 minutes left. I just wanted to maybe stop here. Thank you, everybody, for participating and for your questions. I thank Pina and Dr. Bissonnette for a very good presentation. We got positive reviews right live here. So thank you very much for everybody attending. And as I said, we will follow up with you shortly for anyone that would like to re-listen to the presentation or get a copy of the slides. So have a good rest of day. Thank you very much. Thank you.